Hello, very good evening to the students who have joined the class. Can you please tell me in the chat box if my voice is audible and my video is visible? Also let me know that if you can just see the PPT. Is everything alright? My voice, the video and the PPT also? Okay. Okay, thank you. So we will wait for a few minutes and then we will start today's session. Okay, so very good evening again. My name is Moshu Medev and I am the PMRF Fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. So today it is the week 12 live session for the NPTEL course of Medicinal Chemistry. So I think it is already 6 p.m. but we will wait for a few more minutes so that more people will join and we will have more students in our class. So basically we have completed this all. Okay. Yeah, so as I was saying, like today it is the week 12 live session and we have successfully completed previous 11 live sessions in last 11 weeks, and in those live sessions basically what we did, we did discuss about the course content of each week, and we basically discuss related questions like related MCQs of the courses, so that you can solve the assignments correctly. But since we are done with the assignments all of all weeks and all course content, so today it will be a summary class. So, in this summary class, basically what we will do, we will summarize what we have learned so far about drug discovery, about medicinal chemistry, about and the topics related to medicinal chemistry. There will be few questions today at the around I mean toward the end of the session, but those questions are not is not directly into your course content, but it is somewhat related. So, I thought it would be good to incorporate few extra questions also, but mostly we will today discuss, and I will summarize the course, so that you will just have a look look at the things what we have finished in the previous classes. Okay. So, today we will learn basic medicinal chemistry at first, and one more thing I would like to say, that tomorrow morning from 11 to from 10 a.m. to 11 a.m. I will also have another problem solving class, extra problem solving class. So, and it will be actually your last class of this session, and it will be this 1 hour class. We will basically discuss about related topics like many diseases like cancer, and we will discuss the drug related to the diseases like antibacterial, antiviral, antifungal, antihypertensive, and also anti-cancer, all those things. So, these things we will discuss tomorrow morning class, and today we will have a look at the basic medicinal chemistry. Okay. So, we will look at the drug discovery process, then we will look about something which is called the intellectual property rights or IPR. Then we will move on and we will have some discussions on your therapeutic index, IC50 values, these kind of things which are very commonly used terms in the medicinal chemistry. Then we will have a look at the assays which are used for the medicinal chemistry, and the very important pharmacokinetic, pharmacodynamic parameter. Also, we will have a look around the enzyme kinetics, and I will just recapitulate few things, so that when you are going to read for your final exams, it will be helpful. Okay. So, I think we can start our class, or we can wait for 2 more minutes. So, I think my voice is clear and you can see the PPT, right. So, if you have any problem during the class, if you cannot hear my voice or if you cannot see the PPT correctly, please let me know in the chat box. Okay. And one more thing, if there is some internet disruption, okay. So, due to bad weather, there might be some internet disruption today also. So, if it happens, I want all of you to wait for some time, and I will surely rejoin soon. Okay. And one more thing, if you have any doubt regarding any topic of the class or any topic of your course, you can let me know in tomorrow morning's class or today's class itself also, or you can let me know in tomorrow morning's class. Okay. Or you can just drop an email to me. I have always shared my email ID with you, so you can do it. Okay. You can also put your queries via email, because after tomorrow we won't have this kind of live session anymore. So, it is basically today and tomorrow sessions are the last classes for this course before your examinations, right. So, I think it is time. So, we can start our session. If more students join, we will just add them up. Okay. Okay. So, basically we will just summarize what we have learned so far in the last weeks. Okay. So, the first thing which we learned, I think it was about drug discovery, and in the very important thing about the drug discovery was the clinical trials. So, what was the clinical trials? Like, we are designing a compound to be a drug, okay, and before going to the market, the drugs actually have to go through various examinations. Okay, so the pattern of the examination, so as I don't know it correctly because I am not the official TA of this course, but it will be descriptive, I think, and I think these things like the pattern of the examination and all those details, it will be shared to you soon if it is not done so far, but it will be shared to you soon. I think it is your examinations are from 30th April if I am not wrong. So, I think it will be shared to you the details and the pattern of the examination soon via email, or it will be updated on the NPTEL website, but it will be descriptive. It won't be, I think, from the MCQ pattern, but it will be descriptive. Okay. Okay, so the clinical trials. So, why the clinical trials are necessary? So, the clinical trials are designed to determine safety and tolerance in human for due to the to see the pharmacokinetics parameters, to see the bioavailability for a range of doses, to determine the pharmacological profile. The main phases of clinical trials or preclinical trials are the preclinical animal studies, submission of investigational new drug application to government bodies such as US FDA. So, the phase 1 is the normal and healthy human volunteers, it is tested the drug. The phase 2 to evaluate the safety and efficacy of the drug in the patients. Phase 3 large patient number study to establish efficacy versus a placebo or competitor compound. And phase 4 long term surveillance or monitoring for the adverse reactions. So, these are the few steps for the clinical trials, and this is the flow chart for the drug discovery and development process. So, as you can see, this is the percentage of total R&D or research and development investment per one new drug. Okay, and this is the strategic decision on target indication, and this is in this right hand side it is mentioned the years. I mean how much of the time is needed for each step to be cleared, right. So, the first step is the drug discovery and preclinical phase. So, in this case what happens, it takes nearly 3 to 5 years, as you can see, okay. And as you can see, 5,000 to 10,000 compounds are there at the first stage, at the drug discovery stage, okay. And after that, when it goes to the preclinical study, from that 5,000 to 10,000 compounds, only 250 compounds are taken, okay. And at the end of this preclinical study, only 5 compound, nearly 5 compound are selected for the clinical trials. So, as you can see, not only this process is time consuming, but also it is very much thorough and also very much complicated, because you to eliminate 5 to 10,000 nearly similar kind of compounds is not an easy task, okay. And 30% of the investment goes to this drug discovery and preclinical phase. Here the drugs are designed, the drugs are synthesized, and they go to the preclinical phases. And what is the preclinical and all the clinical stages, I think I have discussed in the your very first class, okay. And after, when these things are cleared, then we will go to the clinical trials. Now, in this clinical trials, total 50% or of this investment goes up, okay. And from this 50% is all again divided into 40-60% ratio, means 2:3 ratio. The first 40% investment goes to the phase 1 and phase 2 trials, okay. And this phase 1 and phase 2 trials, after that, now what happens, like after it goes to the second, then it goes to the phase 3 trial, and it takes up to the 13 years to have a new drug. And then regulatory phase, it takes up to at least 2 years and 20% investment, and at the end of the one FDA approved drugs is arrived in the market, okay. And the phase 4, as I mentioned, phase 4 is nothing but a post market surveillance, means when a drug is approved by FDA to be used in the market, but after some year also another surveillance or another trial is also, it is takes place, where what happens there, they test, they actually test, like when the compound is in market, means when the compound is in the larger population, is there any side effect or is there anything which is problematic related to the drug, then if it is okay, then it is okay, but if there is a problem, the drug is eliminated from the market, okay. So, this is the thing about the clinical trials, the time and the money it is consumed by this clinical trial. So, compound success rates for the clinical trials, 40% of the compounds advance from phase 1 to phase 2, 1/3 of the compound advance from phase 2 to phase 3, and 2/3 of the compounds they advance from the phase 3 to the phase 3 to FDA approval, and 10% of compounds entering the clinical trial stage advance to the FDA approval. So, this is the this is called the inverted pyramid of the drug discovery and development. Okay. Okay. So, now different regulatory bodies. So, there are very different regulatory bodies, and this is also very much related to your okay. So, drug discovery process. So, you should know these things. So, the most important regulatory body is the FDA. And what is the full form of FDA? It is the Food and the Drug Administration. Okay. And it is a federal agency of the United States Department of Health and Human Services, one of the United States federal executive departments. The FDA is responsible for protecting and promoting public health through the control and supervision of food safety, tobacco products, dietary supplements, prescription and over-the-counter pharmaceutical drugs, medications, vaccines, biopharmaceuticals, blood transfusions, medical devices, electromagnetic radiation emitting devices or EREDs, cosmetics, animal foods and feed, and veterinary products. Okay. Similar to this FDA, there is also which is called the European Medicines Agency, Science and Medicines of Health. So, this is the governing body for the Europe. MHRA, it is the Medicines and Healthcare Product Regulatory Energy, and it is the an executive agency. So, MHRA, it is the Medicines and Healthcare Products Regulatory Agency, and it is the executive agency of the Department of Health and Social Care in the United Kingdom, which is responsible for ensuring that medicines and medical devices work and are acceptably safe. There is a PMDA, which is Pharmaceuticals and Medical Devices Agency, and it is a Japanese governmental organization similar to function of the FDA of the United States, or MHRA of the United Kingdom, or Central Drugs Standard Control Organization CDSCO in India, or the European Medicinal Agency of Europe. So, these governing bodies are different regulatory bodies of different countries or different areas. Like for India, there is the Central Drugs Standard Control Organization CDSCO under the Directorate General of the Health Services, Ministry of Health and Family Welfare, Government of India, and it is a National Regulatory Authority of India. And there is also FSSAI, which is Food Safety and Standards Authority of the India. Okay. And we actually have this, we have heard of this because there is a FSSAI, I mean this is actually you need to take the approval of this before taking up the any food thing in the market. Okay. So, these two are the governing bodies of the India. So, I think in the if I remember correctly, in the first class itself somebody has asked like in India do we have any governing bodies like FDA. So, yeah, in India we also have some governing bodies like FDA. I think FDA is the topmost, but India also itself have its own governing bodies like CDSCO and FSSAI. Okay. So, this was about the various regulatory bodies. Now, there is one term which is known as generic drugs. Now, what is a generic drug? So, a generic drugs in short, generics. Okay. So, this is a drug which is produced and distributed without patent protection. The generic drug may still have a patent on the formulation, but not on the active ingredient. A generic must contain the same active ingredients as the original formulation. When generic products became available, the market competition often leads to the substantially lower prices for both the original brand name product and the generic forms. In the US, drug patents give 20 years of protection, but they are applied for before clinical trials begin. So, the effective life of the drug patent tends to be between 7 to 12 years. Okay. So, India is the world's largest manufacturer of the generic medicines. and these are the various pharmaceutical agencies which are the reason because of the India's largest manufacturer of generic disease like Sun Pharma, Dr. Reddy's, Cipla, Luptin, Zydus, Aurobindo, Glenmark, Torrent Pharma, Ranbaxy, Natco. So, these are the manufacturers of the generic medicines in the India. Okay. So, in the drug discovery and development process, one thing which is very important, and I think I have discussed it earlier, I do not remember, but it is the assay. Okay. So, assay and assay development, these are very important for the drug discovery and development process. Now, what are the assay? Assays are the investigative procedures that assess a compound's effects on various biological targets. Assay development involves optimizing the conditions for testing in preclinical setting. These conditions include temperature, pH, salt concentration, solubility, order of reagent addition, buffer and other components required to make the assay functional. No drug discovery project can be successful without a proper assay. The developed assay should be relevant to the disease. The assay should give a reproducible result. So, basically assay is nothing but you can say it is a procedure or it is an experiment to see how much the compound is affecting on a particular biological target. For example, I will just tell you there is one assay which is called a MTT assay. Now, what is a MTT assay? So, basically it is tested to see if the compound which we are designing is cytotoxic to a particular cell line or not. Okay, it is called the MTT assay. So, what is a MTT? MTT is basically nothing but it is a compound. So, this compound is converted to a particular salt. So, this MTT, the color of this MTT, it is this color of this MTT is kind of orangish. Okay. And when this MTT compound, it is converted to a salt, and this salt, the color of the salt is purple. And what is the mechanism by which this MTT is converted to this purple salt? The mechanism takes our cells ATP's energy, basically our cells the energy from the normal healthy cells ATP, this is taken by this MTT compound, and it then the purple crystals of the salt is formed. Okay. Now, how you will check if your compound is cytotoxic or not? So, what we have to do? We have to take a plate, okay, and this is called there is some plate cell culture plates and all things. So, what we have to do? We have to take this compounds. We have to take the cells in those vials. So, I think you can see this picture, right? This picture. So, here as you can see there are some few wells and there is an array of wells. So, in each row we take the same concentration of the cells, and in each row we add the compounds, and few rows there are no compounds is added. Okay. Now, if the compound is cytotoxic to the particular cell, what will happen? These compounds will kill the cells which are in these plates in these wells, and if the cells are dead, what will happen? No ATP will be there. Okay, if there is no living cell, there will be less, the lesser the living cell, lesser will be the ATP concentration, and if the lesser is the ATP concentration, what will happen? Lesser this conversion of this orange MTT to this purple salt will happen. Now, after some time, so what happened, like we have to take the cells in this well, we have to incubate it. After some time we have to add the compounds, okay, and we have to wait for 1 to 2 days. And after 1 to 2 days, what happens, like we just take up the solutions and we add some solvent like DMSO to dissolve the crystals and to see how. So, in the wells where the more purple crystals are formed, or the intensity of the purple crystals color is high, means in those wells the cells were alive and in the most concentration, and if the cells are dead, so the purple color concentration will be lesser. So, by this way we will know if our compound which we have added is cytotoxic to the particular cell line or not. Okay, that's how we actually test if our compounds is cytotoxic. If we have to test our compound, if you want like our compound should not be cytotoxic at all, then we actually use normal healthy cells. Okay, and if you want to test if the compounds are cytotoxic to the cancerous cell, then we actually use the cancer cells for in these wells to culture to check if our compounds can be a potent anti-cancer drug. Okay, so this is the way by which actually we can test if our compounds is cytotoxic or not. So this is a kind of assay. So this was an example only I have said you. So this is one kind of assay. There are many kind of assay, depend on the experiment, depend on the results we need. So various assays are developed. Okay. So a bioassay is an analytical method to determine concentration or potency of a substance by its effect on living cells or tissues. Bioassay are quantitative biological assay used to estimate the potency of the agents by observing their effect on living cell in vivo or tissue or cell culture systems in vitro. Bioassay is used to detect biological hazards or give a quality assessment of mixture. Bioassay is often used to monitor water quality and also sewage discharge and its impact on surrounding. Citation needed. It is also used to assess the environmental impact and safety of the new technology and facilities. So there is another assay which I think many of you have heard the name. It is a ELISA or enzyme linked immunosorbent assay. So it is a quantitative analytical method that measured the absorbance of the color change from antigen antibody reaction. For example, direct, indirect, sandwich or competitive. ELISA is used to measure the variety of substances in human body from cortisol levels for stress to glucose level for diabetes. Okay. So ELISA, I think you have heard about this term ELISA enzyme test very much, because it is a very famous immuno assay. Okay. So this was the example for the assays. Okay. So microsomal stability assay, this is also very important assay for the. Okay, somebody wants to join. Okay. So microsomal stability assay, it is also a very important assay to for the drug discovery process. So a liver microsomal metabolic stability assay was validated to evaluate liver enzyme metabolism using model compounds. In addition, the assay was evaluated by testing a set of structurally unrelated compounds for metabolic stability. Other than efficacy of interaction with molecular target, metabolic stability is a primary factor responsible for the failure or success of a compound in the drug development pipeline. The ideal drug candidate should be stable enough to reach its therapeutic site of action. Despite many recent excellent achievement in the field of computational methods supporting drug metabolism studies, a well recognized procedure to model and the predict metabolic stability quantitatively is still lacking. The metabolic stability of the compounds was assessed in vitro incubations in the presence of liver microsomes and subsequently quantified by liquid chromatography mass spectrometry or LCMS method. So metabolic stability as defined as the percentage of parent compound lost over time is assessed in the presence of liver microsomes. Microsomal stability assay, MLM, RLM, HLM. So this is the MLM is the mouse liver microsome, RLM is the rat liver microsome, HLM is the human liver microsome. So this microsomal stability assay, it actually takes the help of the liver microsomes, and based on the microsomes it is taking from the organism. It can be liver microsomes for the mouse or the rat or the humans also. So this was the microsomal stability assay, which is actually gives us the information about the metabolic stability of a particular drug candidate. Okay. Okay, so in the drug discovery process, there is a very important term which I think used many times, which is called the IC50 value. So half maximal inhibitory concentration or IC50 is a measure of the potency of the substance in inhibiting a specific biological or biochemical function. IC50 is a quantitative measure that indicates how much of a particular inhibitory substance, that is a drug, is needed to inhibit in vitro in given biological process or biological component by 50%. The biological component could be an enzyme, cell, cell receptor or microorganism. IC50 values are typically expressed as molar concentration. Okay, IC50 is commonly used to measure antagonist drug potency on pharmacological research. It is comparable to the measures of potency like EC50 or for the excitatory drugs. EC50 represents the dose or the plasma concentration required for obtaining 50% of the maximum effect in vivo. IC50 can be determined with functional assays or with computation binding assays. Okay. So, IC50 value basically is the concentration of the drug which is required to inhibit a particular biological process by 50%. So, this as you can see this from this graph. Okay. So, in this graph these compounds, this dasatinib, imatinib, ponatinib and nilotinib, these are the which is called the kinase inhibitors. So, I think we have discussed about kinase inhibitors in the last class itself. Okay. So, kinase inhibitor, what the kinase inhibitors do? Kinases are the particular enzymes which actually what the kinases does, they basically takes up the gamma phosphate from a ATP, from the ATP, okay, and it takes up the gamma phosphate and it adds it up to the particular protein. Okay, then where it adds to the actually it phosphorylates the protein and it adds this phosphate group to the amino acids, so which have the free OH group like serine, like tyrosine. Okay, and depending on that it the names of the like tyrosine kinase inhibitor, okay, this kind of serine kinase inhibitor. So, these are the kinase inhibitor type of kinase inhibitor. So, because kinases because from this particular pathway, when a kinase enzyme, it takes up the gamma phosphate from ATP and adds to the your substrate, and this pathway actually is very crucial for the cell signaling for malignant cells mainly. Okay. And if you can inhibit this particular enzyme, so the cell signaling will be inhibited and this kinase inhibitors are due to this the kinase inhibitors are an excellent target for the cancer research. Okay. Many in the after I think 2011 there are till now around 90 kinase inhibitor drugs which are approved by FDA for market use, and these are most the kinase inhibitors are approved due for the different type of cancers, okay, different kind of malignancies. Okay, so like this dasatinib, imatinib, also this so all these compounds are from for any kind of cancers. Okay, like imatinib is used in the RCC, renal carcinoma, renal cell carcinoma, and nilotinib is used for the blood cancer. Okay, so these are the use. So, as you can see, this is the graph which is called the drug response curve. Okay, so like in the last slide I have told you about the MTT assay, right? Like we can check if the compound is active or not by checking how much. So, this IC50 value we can actually plot a graph from the information to like how much concentration is needed. So, there are some formulas and many softwares actually are used to calculate this. So, this is called the drug responsive curve. Okay. In the y axis, as you can see, there is a percentage of inhibition, in the x axis there is the concentration. Okay, and IC50 values, this is the graph and IC50 values are calculated from this graph. So, as you can see, like dasatinib, it has IC50 value of 0.42, imatinib is 474.90, ponatinib is 0.95, nilotinib is 2.85. So, this is for a particular cell line. So, in this cell line from this information, it is find to like dasatinib is the best kinase inhibitor, because the IC50 value in the nanomolar is very less, means at a very lower concentration of the drug it is inhibiting the 50% of the activity. Okay. So, it is the most potent drug from this class of the compound. Okay. So, another term which is very important is the therapeutic index. So, what is the therapeutic index? It is basically the ratio of the undesirable and desirable drug effect. So, for in vivo system, 3 therapeutic index could be the ratio of LD50 lethal dose for the 50% of test animal to the ED50, the effective dose of that produces maximal therapeutic effect in the 50%. Larger the therapeutic index, greater is the margin of the safety of the drug. Okay, so, LD50 is the lethal dose for the 50% of the test animal, and ED50 is the effective dose. So, if the lethal dose is greater than the effective dose, means how the more the lethal dose is greater than the effective dose, the more safe is the drug. Because let us say if the you let us say like you need 70 nanomolar compound as a lethal dose, okay, for the 50% of the test animal, and the effective dose is the 10%. So, what is the ratio? Like 70 by 10, 10 nanomolar, 70 nanomolar by 10 nanomolar is the 7, and another compound, it has the lethal dose is 90 nanomolar, and the ED50 value or the effective dose is again 10 nanomolar. So, what will happen, the ratio will be 9, means the higher therapeutic index means the drug is more safe, because the lethal dose is very high in comparison to the effective dose. So, it is not much of the drug is much safer when the therapeutic index is high. So, these terms like lethal dose, effective dose, ED50, LD50, IC50, EC50, these terms are very much common, and these terms are very much important also. Another term there is for understanding the activity for the kinase inhibitor or any enzymatic inhibitors is the inhibitory constant. Okay. So, there is a term which is called the Ki. So, that is the inhibitory constant, which actually checks how much of inhibition is happens because of the drug. Okay. Okay, let me check. Okay. So, now we will go to a thing which is called the drug metabolizing enzymes. So, we have read about drug metabolism in your course, like the phase 1 metabolism, phase 2 metabolism, all we have discussed. Okay. So, why the drug metabolizing enzymes are important, why the drug metabolization is important. Liver is the major organ for the drug metabolism or elimination. So, there are phase 1 and phase 2 enzymes are there, and in the phase 1 drug metabolism, what happens? Oxidative or hydrolytic reactions happen, and in the phase 2 conjugative reactions are happen. So, predominant enzyme system that metabolizes drugs is the cytochrome P450 or CYP450 family of enzymes, which mediate the oxidation reactions such as hydroxylation. So, what is hydroxylation reaction? It is basically nothing but the enzymes actually helps to take up the OH group from the H2O2 or any water to the particular substrate, or the oxidation reaction can also be the like this, like it takes up the oxygen from the substrate like H2O2 and by converting it into the water and adding up to the substrate. Okay. So, there are this cytochrome P450, this is a family of enzymes. So, there are many enzymes in this cytochrome P450 family, and these enzymes are like CYP1A2. Known drugs metabolized by this enzyme is 4%, CYP2C9. So, known drugs which are metabolized by this enzymes is 10%, CYP2C19, the known drugs which are metabolized is 2%, CYP2D6, known drugs which are metabolized by this is 30%, and the most important one is the CYP3A4, and because 50% of known drugs are metabolized by this particular enzyme. Okay. So, this particular enzyme actually helps us to metabolize 50% of the known drugs. And role of the CYP enzymes in the hepatic drug metabolism, as you can see here, percentage of the drug metabolized by CYP enzyme. So, this is basically the pie chart for the relative hepatic content of the CYP enzymes. Okay. So, this is the flow charts of this related enzyme. So, for an example, So, okay. So, as I have said you, like the important role for this cytochrome P450 is the hydroxylation reaction. Now, how it happens? So, cytochrome P450 are the family of enzymes containing heme or iron as a cofactor that function as a monooxygenases. In mammals, these proteins oxidize steroids, fatty acids and xenobiotics and are important for the clearance of various compounds as well as for hormone synthesis and breakdown. Okay. So, this is a substrate which is, so this is an example how this particular enzyme cycle goes. Okay. So, this is a particular compound which is called the 3-methyl indole. Okay. So, by this P450 enzyme, hydrogen atom abstraction happens at first. So, the hydrogen atom is abstracted from this position, okay, and CH3 it is converted to CH2 dot. Now, this is the Fe4 OH and this is the in the active site of this particular enzyme, and electron transfer happens, okay, and the CH2 dot is converted to CH2 plus, okay, and then there is this from this thing hydration can happen from the, and 80% time hydration happens, and this hydration gives us to this hydroxylated product, okay, or H plus ion can also be eliminated and this 3-methylene indolin can be formed, but this reaction is not that much of suitable because this is I mean like this reaction is not too much suitable, that is why 80% time hydration happens, okay. Hydrogenation of 3-methyl indole to 3-methylene ion by cytochrome 450. So, this was the example. Okay. So, till now we have discussed like about clinical trials, clinical bodies, we have discussed about various governing bodies, about generic drugs, then we discussed about various assays, IC50 value, and also we discussed about the enzyme metabolisms, the drug metabolism. One more thing which is very important, and I think we have discussed it over 2 to 3 weeks, the pharmacokinetics and pharmacodynamics. So, these two terms are two pillars of the medicinal chemistry. These two terms are very much important. So, pharmacodynamics. Pharmacodynamics is the study how the drugs interact with the molecular target, that is the effect of the particular drug on the body. Pharmacokinetics is the study how the drug reaches to the target in the body and how it is affected by on the journey, that is the effect of the body on the drug. Pharmacokinetics is the study of how is the drug is absorbed, distributed, metabolized and excreted in the body. Okay, so basically I have just I think told you in the million times, like the difference between pharmacokinetics and pharmacodynamics is nothing but you have to remember, like pharmacokinetics is what our body is doing to a particular drug. So, we are taking a drug, the drug is absorbed in our body, it is getting metabolized, it is getting excreted. So, this is all of this about like what our body is doing to the drug, but the pharmacodynamics is just opposite, what the drug is doing to our body. So, these two terms are very much important in the heart of the medicinal chemistry when you are discovering a particular compound to be a particular drug. This pharmacokinetics and pharmacodynamics are the things of which you have to take care of. Okay. So, this flow chart is shown here about the pharmacokinetics. So, first is the drug administration. Now, it can be oral, it can be intravenous, it can be intraperitoneal, it can be subcutaneous, it can be intramuscular, or it can be inhalation. Okay. The second step is the absorption and the distribution. Membranes of the oral cavity, gastrointestinal tract, peritoneum skin, muscles and lungs, by which all of these things the drug gets absorbed and distributed. Now, distribution goes to like this. So, it can do the first is the binding. So, what happens, the drug goes to the plasma with the plasma protein binding, and then it goes to the neuron receptor which is the target site, okay, and then after coming back what happened, the plasma protein binding, and it goes to the liver, and from the liver what happened, it gets metabolites, and these metabolites can go to the blood plasma, to the bone fat in the inactive storage deposits, and then also it can go to the for the excretion like the intestine, kidney, lungs, sweat glands, and the excretion product like feces, urine, water, vapor, sweat, saliva. These are the excretion process. So, all of this is the pharmacokinetics parameter, pharmacokinetics, like how the drug is interacting in our, after we are taking the drug in our body, how the drugs are getting going to places and how it works. And pharmacodynamics, so there are receptors, protein molecules with one or more binding sites located on the cell membranes receive a signal from body's chemical like neurotransmitter, hormone and enzyme. Signal will cause the molecular event on the inside of the cell to occur. Drugs enhance, agonist, diminish, partial agonist, block, antagonist, the generation, transmission or receiving the signal. Affinity, the attraction between the drug and receptor. High affinity, the drug will bind easily to the receptor. Low affinity requires a higher concentration of the drug to get a therapeutic response. Drug potency, the amount of the drug required to produce a therapeutic response. Dose responsive curve and effective dose ED, amount of the drug that produces a therapeutic response in 50% of the people taking it. Toxic dose, amount of drug that produces adverse effect in the 50% of people taking it. Therapeutic index, margin of safety, which we discussed just before, like ratio between TD and ED. Higher the therapeutic index, safer is the drug, considered to be in general non-prescription drugs that are higher TI the prescription drug. Okay. Now, there are few parameters which are very important in the pharmacokinetics, which are called the pharmacokinetic parameters. The first thing is the peak concentration or the Cmax value, the peak plasma concentration of a drug after administration. The amount of drug when it reaches to the maximum concentration in the plasma, it is also known as the maximum plasma concentration. Peak time or Tmax is the time when the drug concentration reaches to the maximum after the drug administration. At this point, the rate of the drug absorption is equal to the drug elimination. Terminal half-life or T-half is the time required for the concentration of the drug to reach half of its original value. Area under the curve or AUC is the integral of the concentration time curve after a single dose and or in the steady state. It is a measurement of the extent of the drug bioavailability. It shows the total amount of active drug that is bioavailable in the systematic circulation. Clearance or KL is a rate at which the drug is removed from the body per unit time. So, these are few parameters. These parameters are very important to check how the drug is working, and these parameters are used when there is pharmacokinetic study during the preclinical studies. Okay. So, these parameters or these values are very important, by which we can plot a graph to check how much efficient a particular drug is to a particular target, or for a range of the drugs we also can calculate these things. Okay. So, now one more thing which is very important for your study is the enzyme kinetics. Okay. So, enzymes, how the enzymes works and all these qualitative things we have read so far, but I think you should also know about the quantitative things like the equations, how the equations come, why the equations are important. Okay. So, what is enzyme kinetics? So, the definition, the study of the rate at which enzymes works is called the enzyme kinetics. So, as you can see, these are the substrates, and the substrates they bind to the enzyme. Okay. So, and there is a high substrate and this is the enzymes. Okay. And this smaller spheres are the substrate. Okay. So, there may be high substrate concentration or there may be low substrate concentration. Okay. Reaction rates increase as substrate concentration increase, but they become saturated at very high concentration of the substrate. For a given enzyme concentration and or relatively low concentration substrate concentration, reaction rate increases linearly with the substrate concentration. However, at relatively high substrate concentration, the reaction rate asymptotically approaches to the theoretical maximum. The enzyme active sites are almost all occupied, and the reaction velocity at this point is represented by Vmax. The substrate concentration midway between the two limiting cases are denoted by Km. Yeah, higher the rate therapeutic index, safer will be the drugs. So, so as you have seen, like what is the therapeutic index? It is the ratio between the lethal dose and the effective dose. Okay, so the lethal dose is basically nothing but the how much of the dose is required for this drug to be to kill a particular organisms. Okay, and the effective dose is how much concentration it is needed to be particularly effective toward a particular disease. So, if the ratio is high, means the difference between the lethal dose and effective dose is very much high, means the drug is more safer. Okay. So, yeah, you are right, higher the therapeutic index, safer will be the drug. So, but we were discussing about enzyme kinetics. Reaction rates increase as the substrate concentration increase, but become saturated at very high concentration of substrate. For a given enzyme concentration and for relatively low substrate concentration, reaction rate increases linearly with substrate concentration. However, at relatively high substrate concentration, the reaction rate asymptotically approaches to a theoretical maximum. The enzyme active sites are all almost occupied and the reaction velocity at this point is represented by Vmax. The substrate concentration midway between two limiting cases is denoted by Km. So, this is basically is the enzyme kinetics, and which is the graph of the enzyme kinetics. So, as you can see, all the things which are mentioned here are represented by this graph. So, this is the enzyme kinetic reaction. So, E is the enzyme, S is the substrate. Okay. And at first the step is called the substrate binding step, where the enzyme and substrate complex is formed, and then the step is the product forming step, which is the catalytic step. Okay. And here the enzyme is stays as it is and the product is formed. So, the rate constant for the first forward reaction is K1, and the first step actually it is the reversible one. Okay. So, the enzyme substrate complex can also revert back to the enzyme and substrate individually, and the rate constant is K minus 1. Okay. And then it goes the second step, it is only forward moving, it is irreversible, and the rate constant is the K2. Okay. And this is the graph. So, this is the at the x axis we have the concentration of the substrate. In the y axis we have the velocity or the velocity of the reaction. Okay. So, as I have said, in the lower substrate concentration or when the substrate concentration is very low, okay, for a given enzyme concentration, the reaction is approaching linearly, and here the first order kinetics is followed. Okay. In the middle there is a mixed order kinetics is followed, and at the end when the substrate concentration is very much high, what happens, like this curve goes to the this curve goes to theoretical maximum. Okay. And it remains unchanged, because here all the enzymes are occupied by the substrates. Okay. So, there is even there is a more substrate, there is no enzymes are available. Okay. So, it goes to the theoretical maximum, which is called the Vmax. So, for a simple single substrate enzymatic reaction for conversion of substrate to product is as follows. I have mentioned it. So, K1, K minus 1, K2 are the rate constant for the individual step. A key assumption in this derivation is the quasi steady state approximation, namely that the concentration of the substrate bound enzyme, this is and hence also the unbound enzyme, change much more slowly than those of the product and the substrate. So, this is the quasi state approximation which is taken here. So, as we can see, so now we just have to use the kinetics formula, which is ddt of this ES. Okay, it is equal to 0, means the change is basically if the change of ES is very much I mean it can be stated as constant. Okay. Because the rate of change of this enzyme substrate concentration is very much lower, the change much slowly than those of the product and substrate. So, it can be taken as 0, the derivative, because it can be taken as constant. Then it is equal to K1 ES minus ES plus this, means what happens, like I will just draw from with the pen. So, since this concentration in this quasi-state approximation, what happens, like the concentration of this ES, it remains constant. So, if it is constant, the rate of change what will happen means this ds ddt with time, means the rate of change of concentration with time of this enzyme substrate bound thing, it is 0. Okay. Now, this thing is equal to this. Now, this stand for like the how this K1 E and S means the formation minus the I mean the breaking of this enzyme substrate complex. Okay. And how it is breaking, it can, this enzyme substrate complex can be be broken by two methods. It can also, it can revert back to enzyme or substrate, or it can just go to the enzyme or product. Okay. So, that is how this equation comes, and this can be rearranged to this part. Okay. This is a very easy kinetic solution. Okay. Now, to simplify the equation, we define the Michaelis constant, and the Michaelis constant is very important, and all this thing is called the Michaelis-Menten enzyme kinetics. Okay. So, this Michaelis constant is Km, which is K minus 1 plus K2 by K1. Okay. K minus 1 is this constant, and I have discussed each and every constant, I think. And the so the if we take this equation here, so we can replace this thing by this Km. So, this Es equal to E into a concentration of S divided by Km. So, total concentration of enzyme which can be given to E0 is given by concentration of free enzyme E and the concentration of enzyme substrate bound substrate. Okay. So, using this concentration of the concentrations to the bound and the bound enzyme concentration 1 can be written as this. Okay. So, E can be written as E0 minus Es. Now, in this equation we will just replace this term like this and this rearrangement. So, the production rate is now this. Okay. Now, after all the substitutions you will get the rate of the product formation, which is Vmax into the concentration of S divided by Km plus S. So, this equation is this was all the calculation to show you like how the equation comes, because this is the important, the product formation equation. Okay. Okay. So, what is the, let me remove this. So, what is the understanding the Km and Vmax. So, these two terms are very important whenever you are given, and I think in your examinations also you will be given few mathematics or mathematical questions related to this enzyme kinetics. Okay. So, basically in those mathematical equations or mathematical questions, these terms are given like Km and Vmax. The values are given in the most of the times, and you have to calculate like product formation or anything. Okay, or like this by using that particular equation. Okay. So, Km, what is a Km? Km is the concentration of substrate at the Vmax, half Vmax. Okay. So, it is the constant for a given enzyme. Km is an estimate of the equilibrium constant for the substrate binding to enzyme. Small km means tight binding, high km means weak binding. Km is the measure of substrate concentration required for the effective catalysis to occur, and Vmax is a constant for a given enzyme. Vmax is a theoretical maximum rate for the reaction, but it is never achieved. To reach Vmax, we will require all the enzyme molecules to have tightly bound enzyme substrate, which is not possible. Okay. Okay, so in the previous graph it was the which I have mentioned, like this graph is the enzyme kinetics for the enzyme kinetics, and it is the all these things were the Michaelis-Menten equation related. Okay. So, in this enzyme kinetics few more graphs are very important, and some of the graph which is very important, it is called the Lineweaver-Burke or double reciprocal plot. So, as I have shown you the equation like this. Okay, so this equation. So, if you rearrange this kind of equation, okay, and if you rearrange this thing, if you rearrange the Michaelis-Menten equation, you will reach to something which is called this, and this is called the double reciprocal plot. So, if you just compare it with the, if you just plot 1 by V divided by 1 by S, okay, taking it as y, taking it as a x. So, y equal to mx plus c. So, m is the slope of this line is km by V max and intercept is the 1 by V max. So, in many question it can be it can come in your examination like using Lineweaver-Burke plot. Just solve the exam questions. Okay, it can be given like the slope is this and the intercept is this. Now, find km. Okay. So, if the slope is given and the intercept is given, means you will have the value of V max, and if you substitute the value of V max here, we will know the value of Km. So, this kind of questions can come from this enzyme kinetics. Okay. So, till now we have discussed many things related to your courses, right. So, we have discussed clinical trials, we have discussed regulatory affairs, regulatory bodies, we have discussed about your pharmacokinetics, pharmacodynamics, also enzymes, how the enzymatic reactions happen in the body by the CYP family. We also discussed about therapeutic index, we discussed about the IC50 and the drugs, all those things. Again we discussed about the enzyme kinetics, okay, and the how much how much the questions can become like from this enzyme kinetics, how the calculations happen, how the derivation happens. So, for the derivation part, I will say I think that the derivations are not much important from the enzyme kinetics, but I showed you the derivation because if the maths come from this some tricky mathematical questions come from that part, so that you can solve it. That is why the derivations are somewhat important. Okay. So, till now we discussed these things. So, now I will just give you like every day we will solve 10 MCQ questions. Okay. So, I will give you 5 minutes break and I will first show you the first question, okay, and use this 5 minutes to try to answer the question, and after 5 minutes when I will rejoin. I am not leaving the class, I am here, but I will just keep my camera and microphone off. So, when after 5 minutes I will rejoin, I want all of you to try to answer the question. Okay, it may be wrong or it may be right, but at least you all should try, because it is the last class. I think you should try to answer the question. You may not know the answer, but if you know, you can try to answer this. Okay. So, I will show you the answer, then I will give you 5 minute break. Okay. So, the first question, it says that cyanogen bromide is specific in breaking the peptide bond associated with which amino acid? Your options are number A arginine, number B methionine, number C histidine and number D proline. So, there is a particular reagent which is called cyanogen bromide, and it is used to break a peptide bond associated with a particular amino acid. So, whenever that particular amino acid is there in the peptide chain, and if you are using cyanogen bromide, the bond will be broken. So, you have to identify which amino acid is this. Okay. So, now I will just keep my camera and microphone off, but I am not leaving the class. Okay. So, I think you should okay, there are already answers are here. Okay. So, take a 5 minute break and others also try to answer. After 5 minutes I will show you the correct answer and we will show what is the correct mechanism for the same. Okay. Hello. Are all of you are present in the class? And is my voice audible to you? I have rejoined, and is my voice is audible? Okay. Thank you so much. So, okay, we will just start. So, we have few answers like D, proline, D, C, and B. Okay, so I think we have multiple answers for it, right? Okay, let us check. So, just like every week, today we will also solve few questions and to check, and these questions may not be we have not discussed yet. Okay, so the first question was the cyanogen bromide is specific in breaking the peptide bond associated with which amino acid. Now, the correct answer is number B, methionine. Okay, so I think there was somebody has answered it like methionine. Okay, perfect. So, what is the mechanism? So, this is the formula for the cyanogen bromide. Okay, and if there is a methionine present, methionine mean is it have a side chain of this thiomethyl group like this SME. Okay, this is the side chain of this methionine. So, this is a peptide chain, for example, and it has this methionine present. Now, what will happen? This sulfur, it will attack with this lone pair to this carbon of this cyanide, and this bromine will be eliminated, and we will have this kind of structure. Okay. And what will happen next? The next it will happen like this nitrogen from the nearby peptide bond, it will just give it here by resonance the lone pair, and this oxygen will attack to this place, and this sulfur it will be eliminated. Okay, and we will have this CH3SCN, and we have this kind of 5-member ring in between the peptide bond. Now, what will happen when you are adding some water here? So, it will be hydrolyzed, okay, and there we will have this a homoserine unit, okay, and the C terminal peptide fragment. So, it will be cleaved from here. Okay, as you can see, this RB and RA was here, RB was here, and this due to this methionine, this ring is this chain is broken from this place. Okay, and we have this N terminal peptide fragment and a C terminal peptide fragment. So, this cyanogen bromide, it is used for breaking a peptide bond associated with the methionine amino acid. Okay. Now, next question, it says which amino acid is termed as helix breaker, and your options are number A, alanine, number B, lysine, number C, proline, and number D, histidine. Again, try to answer the question in the chat box. Which of the amino acid is termed as helix breaker? Alanine, lysine, proline, or histidine? It does not matter if your answer is right or wrong, but try to answer. Which amino acid is termed as helix breaker? Okay, let me check if there are any answers. Okay, so the number C, number D, number D, number A. So, we have number C and number okay, so we have number C and number D and also number A as the answers. Okay, so let us check. So, the correct answer is number C, proline. So, proline is the amino acid which is known as the helix breaker. So why? So this is the structure of the proline. So as you know, proline is the only amino acid which is a imine amino acid, not as a because it is a secondary amine group is present here, not a primary amine group like normal alpha amino acids which have a primary amine group, but this proline, it has a secondary amine group present here, okay, as you can see here. So why it is called the helix breaker? So this is the alpha helix. So alpha helix, what it is called? It is called the secondary structure for the protein. Okay, the primary structure is the peptide bond formation. Then the secondary structure, the alpha helix and beta pleated sheets are formed due to the hydrogen bonding between the nearby peptide chains. Then there is a structure, tertiary structure, where the other forces of attractions are actually takes place, like disulfide bond formation, salt bridge formation, hydrophobic interaction, ionic bonding, covalent bondings, and then we have the quaternary or 3D structure for the proteins. So the alpha helix, this is the alpha helix. So as you can see, the NH group from the one acid, it takes place a hydrogen bonding with the another, and that's how this helix is get stabilized, okay. But if there is a proline present, so when the proline is present, what will happen? This kind of peptide bond will be formed, and there will be no amide hydrogen is present here, and if there is no amide bound hydrogen is present, there is no chance of this kind of hydrogen bond formation. So whenever there is a proline is present, the alpha helix will be disrupted, okay, and that's why the proline is called the helix breaker. Now the next question, an example of oxidoreductase enzyme is number A fumarase, number B lysozyme, number C lactate dehydrogenase, number D RNA polymerase. Enzymes we also discussed today, like different kind of enzymes or different kind of the families of enzymes and isozymes. So then one a very important enzyme is the oxidoreductase enzyme. Now you have to find out among these four which is the correct example for this as an oxidoreductase enzyme. Is it fumarase, lysozyme, lactate dehydrogenase or RNA polymerase? Again, try to write it the answer which you think is the correct in the chat box. So, have anybody answered the question? Okay, I think we have very okay, it is good like all of you are answering. So we have answers like number A, number C, number D, and number C. So we have 1A, 1D and 3C. Okay, let us check. So, the correct answer is number C. Okay, very good. So, we had number C. So, lactate dehydrogenase is the oxidoreductase enzyme. So, oxidoreductase. So, what is oxidoreductase means? It is basically a term which is combined by like oxidation and reduction, and oxidation reduction is altogether it is called the oxidoreduction. Okay. So, like these are the enzymes which are the oxidoreductase group of enzymes like oxygenases, peroxidases, reductases, dehydrogenases, oxidases and hydroxylases. Okay. Like the each and every group of family of enzyme have its own work. Like oxygenases, they incorporate oxygen into organic substrates. Peroxidases, reduction of hydrogen peroxide and hydroperoxides. Reductases, they catalyze reduction. Dehydrogenases, they oxidize substrate by transforming one or more hydride ions. Oxidases, oxygen as the hydrogen or electron acceptor, and hydroxylases add hydroxyl group to substrate. So, these are the various families of oxidoreductase enzymes and how they works. Okay. And this is a very important class of enzyme. Okay. Now, next question. So, in Michaelis-Menten equation at high substrate concentration, the rate of the reaction becomes number A, second order with respect to substrate concentration, number B, 0 order with respect to substrate concentration, number C, first order with respect to substrate concentration, number D, 0 order with respect to enzyme concentration. We have discussed about the Michaelis-Menten equation and all of this just in the last slide, I think. So, now I want all of you to answer the question correctly. So, in the Michaelis-Menten equation at high substrate concentration, the rate of reaction becomes second order with respect to substrate concentration, 0 order with respect to substrate concentration, first order with respect to substrate concentration and 0 order with respect to enzyme concentration. So, haven't everybody answered it? B and D both, okay. B, D, A, B. Let us check. So, the correct answer is number B, 0 order with respect to the substrate concentration. So, as I have shown you the graph, so there was nothing was mentioned as the enzyme concentration, right. So, enzyme concentration during the an enzyme substrate concentration, the enzyme concentration we do not change it, right, we keep it as a constant. So, there is no change with the enzyme concentration, it's the enzyme concentration is same at the start and at the end, but we just change the concentration of the substrate. Okay, so at first what happens, like there is a much higher substrate lower substrate concentration, and then we keep up adding the substrate to the same thing, and the substrate concentration it increases, but there is no change in the enzyme concentration. So, the correct answer it will be 0 order with respect to substrate concentrations. like, okay. So, I have shown you this graph before. So, in the y axis we have velocity of the reaction, in the x axis we have substrate concentration, okay. And the first thing, it is the first order. In this here, when the substrate concentration is low, the reaction is first order, follows the first order kinetics. Why it follows the first order kinetics? Because for a particular when there is a low substrate concentration, so what happens, for every substrate there is an enzyme available, okay. So, for every substrate molecule there is an enzyme molecule available, so the reaction order is first order, okay. It actually depends on the first order concentration of the substrate. But what happens when the lots of enzymes are available here, okay. Here the competition of the enzyme molecules happen, because here there is high substrate concentration, most of the enzymes are getting attached with the substrate, so there is a competition here. So, at this place this is the zero order kinetics, okay. Here almost all enzymes are occupied and this is the saturated phase, okay. So, this is the zero order kinetics, it is followed here. So, this is the high concentration of the substrate. So, high concentration of substrate, zero order kinetics is followed, okay. So, now next problem. Next problem, it says that which of the following statement is false regarding the DNA. Number A, all of A DNA, B DNA and Z DNA have right handed helix. Number B, A DNA is more compact than B DNA. Number C, most common DNA is the B DNA. And number D, A DNA has 11 base pairs in a complete turn. So, there are three different kinds of DNA actually, A DNA, B DNA and Z DNA, and this reaction question is actually about this three kind of DNA. So, try to answer the question, and after you try I will just show you and we will see what is the A DNA, what is B DNA and what is Z DNA. So, have anybody answered it? Number B, number, okay, I think number C, number A, number D. Okay, let me check. So, the correct answer is number A. All of the A DNA, B DNA, Z DNA have right handed helix. This is the false statement regarding the DNA. So, whenever there is a true false kind of question, I have always told you, like read the question carefully, okay. There are maximum I have seen many students to get confused and pick the wrong answer whenever there is a true false kind of question. So, the structural forms of DNA. So, based on the structural forms of DNA, there are 3 types of DNA: A DNA, B DNA and Z DNA. So, this is called the A, B and Z form of the DNA. So, these are the, as you can see, these are the structural form of the DNAs. So, what are the differences between these 3? The characteristic, helical sense. So, the A form of DNA is the right handed helix, okay. B form is also the right handed helix, but the Z form of DNA is the left handed helix. The diameter for the A form, the diameter is 26 angstrom. For the B form, the diameter is 20 angstrom, and for the Z form, it is the 18 angstrom. Rise per turn of the helix, for A DNA it is the 28 angstrom, for the B form it is 36 angstrom, from the Z form it is 44 angstrom. Base pair per helical turn, it is 11 base pair from the A form, 10 base pair from the B form, and for the Z form it is 12 base pair. Helix rise per base pair, it is 2.6 angstrom for the A form, then for the B form it is 3.6 angstrom, and for the Z form it is 3.7 angstrom. Base tilt normal to helix axis, it is 20 degree for the A form, 6 degree from the B form, and the 7 degree for the Z form. Glycosyl bond conformation, it is for the A form it is anti, for the B form it is anti, and anti for pyrimidine and syn for purines for the Z DNA. So, these are the differences between this A form, B form and Z form of the DNA. Okay. So, the most common DNA is actually the B DNA, okay, and A DNA has 11 forms in the complete turn. A DNA is more compact than the B DNA. So, these are the correct statement, but the first one, all the A, B and Z DNA have right handed helix, it is the incorrect, because the Z form of DNA has the left handed helix. Okay. So, now we will move toward the next problem. So, next problem, it says that complementary strand of the primary single stranded DNA is formed by number A DNA ligase, number B DNA primer, number C DNA polymerase, and number D DNA helicase. So, during the DNA formation of the double stranded DNA from the single stranded DNA, there are many enzymes are actually involved. Okay. So, you have to identify correctly which enzyme is involved in formation of complementary strand of primary single stranded DNA. Is it DNA ligase, DNA primer, DNA polymerase or DNA helicase? Somebody wants to join. Okay. So, try to answer this question. So, have anybody answered the question? So, I think we have A, number C, number C, number C. So, we have 3 number C and Y number 1 number A, right? Okay, let us check. So, the correct answer is number C, DNA polymerase. So, this is the chromosome. Okay. So, what is DNA polymerase? So, this is the chromosome. From the chromosome, that is how the original DNA is formed. So, this is the original template DNA. The red signifies the adenine, yellow is the thymine, green is the cytosine and blue is the guanine. Okay. So, as you can see, there is a DNA polymerase enzyme. Okay, and this is the lagging strand and this is the leading strand. Okay, the leading strand is basically following the DNA is formed. Okay, so this is the free nucleotides here, and there is also an enzyme which is called helicase, and this helicase actually breaks the helix of this double stranded DNA. The helix is broken by this helicase. Okay, and this is called the replication fork. And the DNA polymerase enzyme, it actually takes up the free nucleotides which are complementary to the broken strand original template and it forms the DNA by this method. Okay. So, this is the correct, we have few correct answers. So, the next question, correct order of steps of the solid phase oligonucleotide synthesis is number A, deprotection of DMT group, coupling and deprotection of base. Number B, coupling, oxidation of phosphorus, deprotection of DMT group. Number C, deprotection of DMT group, coupling and oxidation of phosphorus. And number D, deprotection of DMT group, oxidation of phosphorus and coupling. So, you have to find out what is the correct order for the steps of the solid phase oligonucleotide synthesis. So, have anybody answered it? So, we have few answers, right? A, C and D. Okay, no problem. Sorry. Here. Okay, let us check what is the correct answer. So, the correct answer is number C, deprotection of DMT group, coupling, oxidation of phosphorus. Now, this is the phase for the solid state nucleotide synthesis. Okay. Now, the step 1 is the deprotection of the DMT group. Okay. And what happened, like acid catalyzed removal of the DMT allows for subsequent base addition. Okay. And the second step, it is the coupling step. The DMT protected phosphoramidate is added to the unprotected 5' of using the tetrazole activator. Then this next step is the capping. The unreacted 5 prime hydroxyl is acylated to prevent further chain extension. Then oxidation of phosphate ester to phosphate using aqueous iodine. And the next step is the release cycle. So, this is basically how the phases happen. So, as you can see, this is the protected nucleotide, and first the deprotection happen, and DMT group is a protecting group of the alcohols, and then the another protected nucleotide, it comes here, and by using this activator it is just attached up to here, the phosphorylation happens, and the next step what happens, the capping happens. Okay, the free OH group is then after the capping, what happened, the next cycle it starts. Okay. Now, the next problem, it says what would be the complementary RNA strand for the following coding DNA sequence in the transcription process. The DNA sequence is 5 prime GCG TATG 3 prime. Read the question very carefully. Okay, what will be the complementary RNA strand for the following coding DNA sequence in the transcription process? Options are number A 5 prime GCG UA UG 3 prime, number B is 3 prime CGCA UAC 5 prime, number C 5 prime CGCA UAC 3 prime, and number D 3 prime GCG UA UG 5 prime. Read this question very carefully, then answer. Okay. Okay, have anybody answered this question? So, we have number C, number D, number B. Okay, let me check. So, the correct answer is number A 5 prime GCGU AUC. So, as I have also said I have said, like, read the question carefully. It says the RNA strand for the following coding DNA sequence in transcription process. Okay. So, this is the your 5 prime to 3 prime and 3 prime to 5 prime system. So, non-template strand or coding strand or sense strand is this, and template strands or non-coding strand or antisense strand is this. Okay. Now, it is said like, so if there is a RNA strand, okay, so if it is given here, like it goes to like 5 prime for the DNA sequence. So, the DNA sequence, it is giving you the this strand. Okay, this is the sense strand or non-template strand. Okay, so if this is like 5 prime, it goes to the 5 prime to the 3 prime here. Now, just you have to take it up like this is 5 GCG TA TGU. So, only the A the T, it will be replaced by the U. Okay. So, this strand, it is called the coding strand, and this strand, it is called the non-coding strand or the template strand. Okay. So, in this question it is asked you for the coding DNA sequence is given, means this is the coding DNA sequence is given. Now, if it is the coding DNA sequence, what will be the non coding DNA sequence or the template strand sequence? It will be 3 prime C G C then A T A C and 5 prime. Okay. Now, you have to just convert it for now the RNA strand, it will be like the complementary for this template strand. So, first, since it is coding is given and this is the coding strand and this is the template strand, first you have to you can just what we can do, you can convert the coding to the non coding or the template strand, and then you can just make the complementary strand for the template strand. So, that will be the given RNA strand for the coding DNA. And if the question was given like this thing is important, like this coding, this is important, this word is very important in this question. If the question was given like write down the RNA strand for the non coding or the template strand sequence is given, then the answer will be something different. Okay. So, that is why I have told you like read the question carefully. Okay, there was one correct answer, so it is good. Okay, now next we will move toward the next problem. So, the next problem it says that after unwinding the double helix, the supercoiling of DNA is prevented by the enzyme number A DNA topoisomerase, number B DNA ligase, number C helicase and number D DNA polymerase. I have shown you about the unwinding of the double helix of DNA in the last slide I think, and I think I mentioned about the enzyme. Now, tell me which is the correct answer, DNA topoisomerase, DNA ligase, helicase or DNA polymerase. Is there any answer? Okay. A number D. So, let us check the correct answer. Yeah, very good. So, the correct answer is number A, DNA topoisomerase. So, this is the DNA replication. So, the first step is that in the DNA replication is to separate the strands of DNA using helicase. So, helicase is the enzyme which this is in this blue portion. So, what it does, it actually separate the double stranded DNA. Okay. Then DNA primase comes to create an RNA primer. Okay. So, DNA primase comes and creates an RNA primer like this here, and this is the DNA primase and this is the RNA primer. Then DNA polymerase comes in and synthesizes new strand of DNA. Single strand binding proteins make sure that two strands do not come back together. Okay. Topoisomerases help to revive the tension, creating the unwinding of DNA. Finally, DNA ligase kicks in and sticks the Okazaki fragments together in the lagging strand. Okay. So, basically what is the role of the topoisomerase? So, I think if you have seen there is if there are two threads which are coiled together, you can just check it by yourself, like taking two threads, somewhat some little bit thick threads, and you just wind it like the double helix like DNA. Then if you try to unwind, then there will be a coiling of the coiling, and there will be due to some tension. Okay. The role of topoisomerase enzyme is to just relieve the coiling, so that smooth DNA replication can happen. Okay. So, in the DNA replication many enzymes are involved. I think you have noticed DNA polymerase is there, DNA ligase is there. Okay. There are there is topoisomerase helicase. So, it is very important to remember the role of each and every enzyme during the DNA replication. Okay. Now, next question, it says that which of the following statements regarding NAD plus is incorrect. Number A, it contains a nicotinamide ring which accepts hydride ion during reduction. Number B, it reoxidizes FAD plus DH plus if it reoxidizes FADH2 to FAD in the last step of the acyl CoA synthesis. Number C, it contains ATP as a part of its structure. Number D, it contains diphosphate linkage. So, NAD, FAD, these are very important in our biological system. Okay. So, this question is again it is a true false kind of question. So, read the options carefully and it is very easy. So, I want all of you to try to answer the question. Okay. Okay, so have everybody answered it? Okay, so we have answers like the screen is not visible. So is the screen was the screen not visible? Is it visible now or is there any problem? Hello? Is my screen visible? Is the PPT visible to you, all of you? Okay. Okay, so we have all the answers like D. So let me check. So the correct answer is number C. It contains ATP as a part of its structure. It is the incorrect statement. Okay. Now let us check what is the structure of the NAD plus. So this is the structure of the NAD plus. Okay. So it contains a nicotinamide ring. This is the nicotinamide ring which accept hydride ion during the reduction. It reoxidizes the FAD plus DH plus to FAD in the last step of acetyl CoA synthesis is also correct. It contains ATP as a part of its structure, which is incorrect. Okay, it does not contain ATP as a part of its structure. So if you look at carefully, this is the structure which may look similar to ATP, but it is not a ATP. So it is it contains ATP as a part of structure is the incorrect. It contains a diphosphate linkage. It is the correct answer because here we have a diphosphate linkage. Okay. And this is how the NAD plus and NADH they differs by there is a nicotinamide ring, it accepts a hydride, and convert this, that's how NAD plus is converted to NADH. Okay. So this was the last question. Okay. So today we actually, okay, I think the for Usha it is the internet connection. I think from your side it was some problem, because I think others you could see the PPT, right? For others, was it the PPT is not visible? Yeah. So I think it is your part the internet was not stable enough, but don't worry. In today's lecture, the PPT and with this lecture video also, I will also upload in the NPTEL website, and it will be communicated to you. Okay, so that whenever there is internet disruption and whenever there is any problem, if you have missed anything by chance, so you can check it from the link which will be provided to you. So today's lecture class recording and the lecture material, the PPT which I have shown in the class, it will be actually this, it will be to you. Yeah, the link will be same for tomorrow's session. So I will remind you one more thing. For tomorrow we will have a 1 hour class from 10 am. I am just writing it here so that you remember, from 10 am to 11 am. Okay. So from 10 am to 11 am we will have another class, and it will be the last class of this session of medicinal chemistry. Okay. And this, in that class today, just like the before starting the question answer session, I just give a brief about the course of medicinal chemistry. So tomorrow also we will discuss the details of medicinal chemistry. Tomorrow mainly we will discuss about antibacterial drugs, antiviral drugs, antifungal drugs, antihypertensives, and this kind of drugs we will discuss about, and the different classes of the drug and all, and we will discuss I think very few questions, I mean like 4 to 5 questions only, and it will be only 1 hour class. It will be the last class of this medicinal chemistry course. Okay. So today I tried to just summarize few things up. I don't know if it will be helpful to you or not, but I tried to cover most of the things which I think I thought it should be taught to you more in more detail, which was not included in the quizzes and all. Yeah, when we will give the admit card of the examination, so don't worry, it will all be communicated to you by the NPTEL website in the due time. So I think your examination is on 30th April, right? So when is your examination exactly? You have got the dates of your examination, right? 30 April. Okay, so before 30 April you will definitely get the your admit card. So today it is 22nd. I think by tomorrow or by Monday you will get your admit cards and all the required details for your examination, how the examination will happen, what is the things which can be come in your examination, and what is the type of the questions and what is the pattern of the question, all it will be communicated to you by tomorrow or by Monday. Okay, so don't worry about it. So tomorrow the class joining link will be the same, which is link of the today's session, and I think it is communicated to you by the email also, the link. So the link will be same, the time will be changed, the time will be from in the morning 10 am to 11 am. Okay. So I think if you have any doubt, any query regarding today's class or anything in general for your course of this medicinal chemistry, you can tell me tomorrow morning. Okay. Also what you can do is, I will share my email ID. So if you have any query, okay, so you can just let me know. One second. So if you have any query or anything, you can just drop a mail to me by tonight. What is the topic you want me to discuss tomorrow? If there is any particular topic in which you think you need more insight, so you can just mail that topic to me tonight, and I will just try to include that part also in tomorrow's class. Okay. So and also if you have any query or any doubt regarding today's class, you can tell me via email, or you can tell me tomorrow. So I want all of you to try to join tomorrow's class as well, because it will be the last class of our for this semester for this course of medicinal chemistry. So I thank you all for joining today's class. I hope you enjoyed the class, and let me know if you have any problem or any doubt. Okay. So we will meet tomorrow morning, and it will be a last class. So I want all of you to try to join the class, because so that we can discuss more topics, because tomorrow's class it will be interesting, because we will discuss a range of therapeutic things which are very important and very interesting part of medicinal chemistry. Okay. So I thank you all for joining. I will close the session for today, and let us meet up in tomorrow morning. Okay. Thank you so much. Thank you all of you.